Hi, everybody. Welcome to the 2020 Komen Virtual MBC Impact Series. I'm Pam Cole, and I'm director of the Komen Metastatic Breast Cancer Collaborative Research Initiative here in the Triangle. The Komen NCTC team and I are so proud to be hosting this session on young researchers in immunology as part of the virtual MBC Impact Series, which in partnership with the Komen Puget Sound, Komen Charlotte, and Komen Atlanta. If you missed the Komen Charlotte's presentation on quality of life issues with 3 metastatic patients and Dr. Chase Bailey Dorton, or any of the previous sessions, the recordings can be found here in Whova by visiting the home page, the home tab to the left of your screen. Each session recording and corresponding documents are linked under the event descriptions. But a word of caution, if you click on any of the other areas of Whova during this presentation, you'll be disconnected from the audio and will need to refresh the page in order to rejoin us. I'd like to thank our local sponsors, Blue Cross and Blue Shield of North Carolina, and UNC Health for their incredible support throughout the year, and to our national sponsors, Seattle Genetics, Pfizer, Lilly Oncology, and Walgreens. And if you're active on social media, please share your thoughts and your takeaways from today's program with your networks using the hashtags MBC Impact and Finding Solutions. Like many of you in attendance, MBC is personal to me. I've been living with metastatic breast cancer for 4 years, and I'm lucky to still be on my first line of treatment. As we all know, the treatments are going to be with us for the rest of our lives, and staying on our first line is so important. I was diagnosed as a stage 1, low risk for recurrence, estrogen positive breast cancer patient. And after 5 years of endocrine therapy, 3 years later, I was diagnosed with a recurrence exactly 4 years ago this month, and ultimately diagnosed with stage 4 metastatic breast cancer. And as we all know, those of you who are caring for people with metastatic breast cancer, if you're living with metastatic breast cancer, what we deal with every day and every week is what does our future hold? And we live with anxiety and wondering at what point is our metastatic breast cancer going to progress? Because at this point, we know that it will. I have my next scans in 10 days, and I'm already starting to feel that anxiety. And it's one of those strange things when you live with metastatic breast cancer, how much we look forward to the scan because we want to know what's going on, and how much we dread it at the same time. But the most important thing that we deal with every day is the need for research. We know that the only thing that's going to save our lives is going to be research that will find the treatments and find the cures for metastatic breast cancer. And I know, like you, I would be happy with a chronic disease. I obviously want a cure, but having a chronic disease would be okay for me and my family as well. And the only way we're going to get there is through research. And we are so lucky here in the Triangle to have amazing research being done right here. It's research that gives me and my family and many of your families hope, as do the incredible researchers we'll hear from today that represent Duke University School of Medicine and UNC Lineberger Comprehensive Cancer Center. It is these two phenomenal research institutions and their close proximity to each other, 8 miles apart, that led our Komen NC North Carolina Triangle of the Coast affiliate to launch the new Metastatic Breast Cancer Collaborative Research Initiative in December of 2019. The initiative will foster collaboration between these two amazing institutions, and it will fast-track innovation toward finding the cures for MBC. Only research projects where Duke and UNC researchers and scientists work together collaboratively on a research project will be considered, and we intend to award our first grant funding in fall of 2021. Visit our Komen, nctc.org to learn more about the fund and how you can help support the fund. Dr. Crosby received her PhD in immunology from the University of Pennsylvania, where she studied activation pathways that regulate immune responses and pathology observed during chronic infection with Drs. Philip Scott and John Wherry. She completed her postdoctoral training with Drs. Kim Lyerly and Zachary Hartman at Duke University. Her postdoctoral work has focused on applying this knowledge of immune activation to the development of novel immunotherapies for the treatment of breast cancers. She was the recipient of a Susan G. Komen Postdoctoral Fellowship Award in 2017 to study a HER2 targeting therapeutic vaccine and the combination of this therapy with immune checkpoint inhibitors. Please join me in welcoming Dr. Erika Crosby to the screen.
Thank you, Pam, for that introduction and for the invitation to talk to you all today about immunology and try to provide an overview that will allow you to understand better what types of immunotherapies are being deployed to help treat your cancer. So I recently read a joke in The Atlantic that made me feel very seen as an immunologist, and I thought it might provide some context for our discussion today. So an immunologist and a cardiologist were kidnapped. The kidnappers threatened to shoot one of them, but promised to spare whoever has made the greatest contribution to humanity. The cardiologist immediately says, well, I've identified drugs that have saved the lives of millions of people. Impressed, the kidnappers turn to the immunologist and ask, well, what have you done? The immunologist says, well, the thing is, the immune system, it's very complicated. The cardiologist cuts him off and says, just shoot me now. So with that context, keep in mind, the immune system is very complicated. Right now, there are about 9 billion immune cells circulating in your blood, with approximately 20 billion more in your skin and gut. And they're constantly surveilling your body to look for any insult that needs to be addressed. So for today, we're going to focus on the 2 main arms of the immune system. These are 2 different types of cells that are going to function slightly differently. So the first is the innate immune system. The job of these cells is to look for common patterns that are shared by pathogens or danger signals or something that's not specific, but it is conserved. So your body knows if it sees this, there's a problem that needs to be addressed. The second arm of the immune system is the adaptive immune system. These types of cells are highly specific, and these are the types of cells that can remember a previous pathogen. So if you hear about immunologic memory, this is the type of cells that are going to get better and better at responding to things as you get older and have seen more different types of infections. So each immune response has several different phases. So for today, I've broken it down into the 3 R's of an immune response. The first thing that your immune system has to do is recognize that there's a problem. The second is they have to mount an appropriate response that's going to address that problem. And the third, which is very important but usually forgotten, is that they have to regulate that response. You don't want to continue responding to that infection after it's been cleared. So you need to shut down the immune response to keep your body from hurting normal tissue. So the first R, recognize. The cells of your innate immune system are mostly responsible for recognizing if there's a problem. So these different types of cells are going to constantly be sampling their environment to look for any kind of danger signals or patterns that are only found in pathogens like bacteria and viruses. So the specific types of cells are called macrophages, dendritic cells, and neutrophils. So these cells are very important in recognizing if there's a problem. Now, it's very easy to understand how these cells would recognize a virus or a bacteria. This is something that comes from outside of your body and enters your body. But we're trying to think about the immune system as it relates to cancer cells. So cancer cells are actually just your body's normal cells that have become mutated and turned into an abnormal cancer cell that divides unchecked. So your immune system knows it can't respond to normal cells. That would be a big problem if your body started fighting itself. And that's actually what happens with autoimmune diseases. So auto meaning self is when your immune system attacks normal cells, and it causes all sorts of problems. The issue is cancer cells aren't really normal, are they? So how does your immune system know that it's okay and even encouraged to respond to a cancer cell? So that's where immunotherapies really come in. So one function of an immunotherapy is to educate your immune system so that it knows when it recognizes a cancer cell that it's okay to respond, that this cell is no longer normal. So once they've recognized that there's a problem, you have to respond. And there are several different types of cells in your immune system that respond. And these are mostly from the adaptive arm of the immune system. So the first cell type is called a B cell. B cells make these proteins called antibodies. An antibody is shaped like a Y. You can see the little cartoon drawing here. The top of the Y is going to recognize a protein on the surface of the cell. So it's going to stick to the surface of a cell very specifically. It'll only stick to that one protein. It won't stick to any other proteins. And then the stalk of the Y is going to stick to another type of immune cell. And it's going to tell that immune cell, look, this is a problem, you need to respond. So B cells are going to make these proteins and secrete them into your body, and they're going to be circulating around, and whenever they stick to a cell, it's going to act like a flag to let the rest of the immune system know, hey, look here, look here. The other type of adaptive immune cell are T cells, and there are two different flavors of T cells. The first is called a CD4 T cell, or a helper T cell. So the job of these T cells is to really provide encouragement and context. So every type of immune response is somewhat different. So if you're responding to a virus, you want one type. If you're responding to a, say, parasite, you want a completely different type of response to help clear that infection. So CD4 T cells are going to provide that instruction to the rest of the immune system to make sure that you generate the type of immune response that's going to best fight that infection. CD8 T cells are my favorite kind of cells. That's what I spend a lot of time studying, so I'm a bit biased, but these types of cells are called cytotoxic or killer T cells. So these are your immune system ninjas. They are going to take all of the context that's been provided by the CD4 cells and the targets that have been labeled by CD8s and identified by all of your innate immune system, and they're going to actually kill them. So these are the types of cells that we want to give the green light to kill off all those tumor cells. So now we've recognized and responded, but now we've got CD8s running around killing things, and we need to regulate this response. So there are these things called immune checkpoints. So these are sort of like the brakes on your immune system to keep your body from damaging healthy tissue. Now, actually, when you have, say, the flu, and it's like 2 or 3 days, you have a fever, you're achy, your head hurts, those symptoms are actually not caused by the flu virus. that's what it feels like to have your immune system fully activated. So you don't want that to be happening for the rest of your life. You don't want that to be happening for even one day longer than it needs to. So these immune checkpoints are very important. The problem is that cancer cells have hijacked many of these checkpoints. And so they express really high levels of all these checkpoints. It's like walking around with a big stop sign. So even if your immune system has recognized the cancer cell and provided the context that it needs to mount an immune response, once the ninja cells get there, they see a big red stop sign and they don't do anything. So that's another type of immune therapy that is pretty recent and is becoming more widely used, are drugs that are going to target these immune checkpoints that are expressed by cancer cells and block them so that the immune system can continue responding. So for my final slide, I just wanted to sort of summarize the types of immune therapies that I'm sure you've heard about or you might hear about later today in other people's talks. And there are many other types of immune therapy. I've just picked sort of 3 broad categories. The first are these checkpoint blockade inhibitors. So as I was just saying, these drugs are going to really cut the brakes on the immune system to allow it to keep responding so that it can fight off those cancer cells. Now, as you can imagine, these brakes are pretty widely used. So there could be side effects. There might be some autoimmune diseases that are induced. But in general, what the research has shown is that we can pretty specifically target these to the tumor and really allow that immune response to take back over and fight off your cancer. The second type is a vaccine. So you're probably very familiar with how vaccines are used for infectious disease. For cancer, what we do is after you have a tumor and we know something about that tumor. So for instance, if you're HER2 positive breast cancer, we can give a vaccine that targets HER2 specifically. So what this is going to do, it's going to take that HER2 protein and it's going to show it to your immune system and say, look at this, go find it. And then it's going to allow your immune system to find everything that expresses HER2 and to attack it. So this is a way of re-educating your immune system to get around that self, non-self. Is this normal? Is it not? And it says, yes, this is not normal. Go after it. The third type of immunotherapy are just antibodies. So we can produce these antibodies in the lab and they can stick specifically to tumor cells. So instead of relying on B cells to know what types of antibodies to make, we just make a boatload of antibodies that say recognize HER2. So Herceptin is an antibody therapy that recognizes HER2 on tumor cells and lets the immune system know that's a tumor cell, this is something we should be responding to. So these are just some of the types of therapies that we're working on to really re-engage your immune system in fighting your cancer. And I think that this is the type of therapy that's going to get us much closer to Susan G. Komen's goal of a cancer-free world. So I'd like to thank you so much for listening, and I really hope that I can address any additional questions that have come up. Thank you.
Thank you, Dr. Crosby, for the presentation on immunotherapy. We are so lucky. We feel so honored every day to be able to work with young researchers like yourself. And for those of us who are living with metastatic breast cancer, we know that the pipeline of science and researchers out there, we need to continue to support because you're giving us hope that you're the ones that are going to find the treatments and cures for us. So thank you. We look forward to hearing your responses to the live Q&A shortly. If you haven't already, please submit your questions to the question and answer section in Whova so Dr. Crosby can answer them shortly. Now, please welcome our moderator, my mentor, my inspiration, Katrina Cook. Katrina has been living with metastatic breast cancer for 9 years and is the facilitator of our Komen MBC support group here in the Triangle.
Hello, everyone, and thank you for having me today. Pam, thank you for that introduction. So as Pam said, my name is Katrina Cook. I have been living with MBC for almost 9 years. It will actually be 9 years in January. I am a triple positive patient and have been doing quite well on treatment for quite some time. Several years ago, I decided that I wanted to help other women like me to not feel so lonely, and with the help of Susan G. Komen, was able to start a metastatic support group. And before COVID, we were able to meet in person and meet live, which was a wonderful thing. Since then, we've been meeting virtually. We would love to have any of you that are listening today, or anyone that you know of, to join us at any time. You can email Komen and we can get you that information. It's the third Thursday of every month, and we'd be honored to have you join us. I'd like to thank Dr. Crosby for discussing the immunotherapies with us, and we do have a few questions. I believe she's about to come back on the screen, so we can ask her a few of the questions that we have. Hi. Thanks so much for your talk. That was great. So we are going to ask a couple of questions that we have gotten. And of course, you know, I've thrown a couple of my own in here too, just listening to you talk, because it's so fascinating and so exciting to those of us living with MBC. And so Marissa says, can multiple types of immunotherapy, vaccine blockers, etc., be used collaboratively on the same patient, or only one at a time?
Yeah. So combination therapy, that's a great question. And the answer is that we're still learning. So combination therapy is going to be the most effective way to use these immunotherapies. As I said, it's complicated. So it turns out when you target one pathway, the tumor is very good at pivoting and using a different pathway, or shutting down a different type of a response. So using a vaccine might be really good at educating your immune system against the vaccine antigen, like HER2, as I used as an example, but the tumor might still have all those stop signs up. So it might not matter how many T cells you have that could respond to HER2 unless you get all those stop signs to come down. So combining an immune checkpoint antibody with a vaccine, we're actually at Duke enrolling a clinical trial right now that is combining a HER2 vaccine with a checkpoint inhibitor to show that that is going to be a really beneficial combination to take advantage of both of those pathways at the same time.
Great. That sounds exciting. Thank you. So Kelly has a question. She has said, thank you, Dr. Crosby. How can we decrease the off-target effects of immunotherapy, given that many of the targets are present on cells other than cancer cells?
Sure. Yeah. So this is where I think it's really important to continue to do a lot of preclinical work. So oftentimes in drug development, you spend a decade or so testing preclinically before it ever gets into patients. And cancer immunotherapy is one of those things that is so exciting and so cutting edge. A lot of these therapies are getting into patients very quickly, which is great. But when we start thinking about cancer as what it actually is, which is a whole bunch of different types of disease, it's not one thing. You know, even within breast cancer, you have your ER/PR or your HER2 or your triple negative, and they all behave a little bit differently, which means patients typically respond differently to these different therapies. So I think it's really important that we continue to study these combinations and model them really well preclinically so that we can do a better job of predicting which patients it's going to benefit. So some of the side effects we won't be able to avoid. If we do our job well, there's going to be some off-target effects. But what we need to know is that the patients who have to put up with those side effects are going to receive a benefit that's much greater than the side effects that they also have. So I think the more that we understand about how these combinations are working, the more we understand about which patients respond and which patients won't. I think that we'll be able to better balance the pros and cons of these types of therapies.
This is kind of just a broad question, but how would the oncologist and patient work together to determine if they are a candidate for immunotherapies?
Sure. So that's a really great question. And I think that we're learning more every day as to what would make you a good candidate. And so there's a lot of genetic testing that's being done on your tumor that could give us some insights into whether or not it expresses things like these checkpoints. So a lot of the early trials showed that if you expressed something called PD-L1, if you were PD-L1 high, that would make you a good candidate for a inhibitor against that pathway. It turns out maybe that's true, maybe it's not, and we need to keep studying it. So I think that there's still a bit unknown and a lot of clinical trials going on. So I think that this is also a great time to just say we're so grateful to the patients that are enrolling in these clinical trials and allowing us to study the genetics of their tumor and do imaging of their tumors to figure out why they are responding, so that in the future we can tell people, you know, you're a good candidate for this or not. So I think this question also gets into access. You know, if you're a patient at Duke or UNC, it's likely that your provider is going to have a pretty good grasp on what's going on and what's available for you and what might be good. But like my grandmother had lung cancer in middle of nowhere, Pennsylvania, and her provider didn't know any of these new drugs. So I think that there's an opportunity to help educate yourselves about it as well.
So when it comes to vaccines, so vaccines and cancer are obviously fairly new and not something that we as patients necessarily know a whole lot about. So if we're talking about MBC specifically, and then about just in general breast cancer at any stage, how do these vaccines work? Or would they be as a replacement for a type of, you know, another type of treatment, or just how do those things work together?
Yeah. So currently we would be using them as a therapeutic that's going to be given with your current therapies. For the example that I can give specifically, we're in the process, we just opened a clinical trial for a vaccine that targets some mutations that occur in estrogen receptor. And we target these because they we only see them in metastatic ER positive cancer. So in your primary tumor, the estrogen receptor isn't mutated in this way. So by giving you this vaccine against the mutated version, we're trying to cut the tumor off at the pass. And if we don't ever allow those mutations to occur, the theory is that we may be able to stop or slow the metastatic spread. So we would want to give you that vaccine before you had evidence of metastasis to really try to cut it off at the pass. So that's just one example, but depending on what we're targeting, it would depend on when the best time to give it would be. And really, all of these things are going to be used in combination. So the goal would be that this vaccine would be able to allow you to go off of other therapies. But as we're testing them, it's just going to be a plus, you know, like maybe this is going to really help get you over the edge or get you, 15, 20 more years.
We have a question from Yvonne, and she has asked, is immunotherapy studies just for HER2 positive? What about HR positive patients?
Yeah, so that's a great question. I know I study HER2, so I tend to go to specifics there. But there are a lot of immunotherapies for other types of breast cancer, hormone receptor positive as well as triple negative. And I know she asked specifically about hormone receptor positive. So as I said, we do have this estrogen receptor vaccine that we're working on. There are also another class of inhibitors that I didn't talk about that target specifically some pathways that regulate how cells divide, and CDK4/6 inhibitors that seem to be really good in hormone positive breast cancers. One of the issues with hormone positive is that they tend to have fewer T cells in general. So something like a vaccine is very promising for that. There's some plasmid vaccines where instead of targeting a specific thing, we use something that stimulates the immune system in general, and we can inject it directly into the tumor. And then that's going to sort of set off danger signals within the tumor so that the immune system is like, hey, what's going on here? Why is there all this danger signal? And start to respond to the tumor again. So there are a lot of options for other types of breast cancer as well. So yeah, no matter what type of breast cancer you have, I think that there may be something that could help you.
Great. And then I have one more quick question before we wrap up and move on to the videos. If this is a question for Angela, if you have received an immunotherapy on the clinical trial, does that exclude you from future immunotherapy trials?
It certainly would depend on the specific inclusion/exclusion criteria of the trial. But as a general rule, it won't always. I know many of our patients, we had a patient who had this was her second type of immunotherapy that she tried, and she went on to do a third type of immunotherapy. And we actually think that the combination that she happened upon was very positive, and we saw, regression of scalp metastases and a lung met following that third line of immunotherapy, and it was 3 different trials. So it certainly doesn't always keep you from being able to enroll, but I can't speak specifically to a certain type of trial. Everyone has their own inclusion and exclusion criteria. But yeah, I think that there's opportunities to have multiple lines of immunotherapy.
Okay, great. I think it's about time that we move on and hear from our young researchers and the exciting things that they are doing now. So Pam, I believe I'm kicking it back to you so you can introduce our young researchers.
Thank you, Dr. Crosby, for sharing your expertise with us today. Your work inspires all of us living with metastatic breast cancer, including Katrina and myself and our families. What you do every day gives us hope for our future. As Dr. Crosby mentioned, there are several facets of immunotherapy, including antibodies, vaccines, and blockers. We are so honored and proud to have research being done on several of these areas right here in North Carolina. We will now hear from 3 local researchers on their studies into these different aspects of how they can be used to treat metastatic breast cancer. First up is Alina Hamilton of UNC. Then we'll hear from Dr. Zachary Hartman of Duke, and Dr. Susanna Garcia will close us out. If you have questions or questions arise during these videos, please hold on to them, as you will have the opportunity to learn more and ask researchers live in the breakout rooms. We're very excited about this opportunity for you to meet and talk to our researchers.
Hello everyone, my name is Alina Hamilton. I'm a 4th-year graduate student at UNC in the laboratories of Katie Hoadley and Melissa Trauster, and it's such a pleasure to be here with you all today remotely. Today I'm going to talk to you about some of my research, which is focused on characterizing the immune microenvironment of breast cancer in Black and non-Black women. So as we've all learned today, tumors are not just made up of tumor cells. In fact, growing evidence has shown that immune cells within tumors, also called the immune microenvironment, play an important role in fighting cancer, and we now know that these cells can be targeted therapeutically to better treat many types of cancer. We're still learning about this in breast cancer, but excitingly in 2019, the first immunotherapy was approved for the treatment of metastatic triple-negative breast cancer after results from the Impassion 130 clinical trial shown here demonstrated that the addition of the immunotherapy drug atezolizumab reduced the risk of progression or death by 40% compared to chemotherapy alone. While this was a wonderful finding and great progression in breast cancer immunotherapy, triple-negative breast cancer, while the most aggressive type of breast cancer and certainly important, only makes up 15 to 20% of all breast cancer cases. And so we still need to learn about how we might be able to target the immune microenvironment and other breast cancer subtypes. In addition, when we look at the demographics of the studied population in this trial, we can see that only 5.8 and 4.9% of the treatment populations were comprised of Black participants. This is particularly problematic because Black women are more frequently diagnosed with triple-negative breast cancer and also have higher breast cancer mortality rates than non-Black women. Given the poor clinical outcomes observed among Black women with breast cancer, other study contexts that do represent these women are really important to further understand these disparities. To address this problem, the Carolina Breast Cancer Study, or CBCS, is an epidemiological study designed to better understand the complex interaction of biology and access to care that drive breast cancer racial disparities. This study recruited over 5,000 women from 44 North Carolina counties and oversampled Black women so that we can get a better picture of the factors that drive disparities in breast cancer. While the CBCS is not a clinical trial, this study is rich in epidemiological, demographic, and biological data, including gene expression now completed on tumors from 1,030 Black and 924 non-Black women. The goal of my work was to leverage the CBCS to characterize the immune microenvironment in breast cancer and to evaluate how it varies with regard to clinical and epidemiological variables, including race and age. To do this, I measured 50 immune-related genes on nearly 2,000 tumors and used clustering to group tumors based on similarities in their immune gene expression patterns. And so on the left, we have a heat map of the gene expression showing this, where tumors are organized in columns and genes in rows, and where red represents the highest gene expression levels and green the lowest gene expression. And we can easily see here that three main immune expression patterns are emerging, with plenty of variation in the types of immune cells that these genes are associated with. And interestingly, when we also looked at the overall immune gene expression levels, we found that these three groups were also defined by an overall low, intermediate, and high immune gene expression, which was how we named these immune groups. We next looked at how these immune groups associated with multiple patient and clinical tumor characteristics, which are listed here on the left. And to break this plot down, I've marked in red the characteristics that are represented by the points here. And when these points are shifted to the right of this dotted line, that means that it's enriched for that specific feature. And so what you can see is that tumors from young women were more likely to be immune high. Tumors from Black women were more likely to be immune intermediate or immune high, and high-grade tumors were more likely to be immune high. While when we looked at tumor stage and node status, we really didn't see strong associations with any type of immune response. And so what this means is that young women, Black women, and women with high-grade tumors are the ones that have these really robust immune responses. And this is the group that we need to focus on to understand how they might respond to different immune targeting agents. And so in conclusion, we identified three immune groups among 1,952 breast tumors in the Carolina Breast Cancer Study that related to an immune high, immune intermediate, or an immune low microenvironment, and that varied based on age, race, and tumor grade. We are now working on investigating how different types of immune cell markers vary across race and tumor subtype, which will be important to help clinicians close the disparities gap in breast cancer. I'd like to thank the CBCS participants that made all of this work possible. And I'd also like to acknowledge the members of the Hoadley, Chauster, and Love Labs that helped me with this work, my thesis committee for their guidance, and our funding sources for supporting this research. Thank you so much for your time.
Hello, I'm Zachary Hartman. I'm a professor in the Department of Surgery at Duke, and today I'm going to talk to you about breast cancer vaccines, a new approach for tumor immunotherapy. So Dr. Crosby's given some background about immunology and cancer, so I want to review the components of a cancer vaccine. There are lots of different components. The first and probably the most important is the antigen. That's the target for the cancer vaccine. It could be a whole tumor, which is a lot of different targets. A protein antigen is one specific protein that the vaccine is designed to target. or an antigenic peptide is really a piece of that protein antigen that the immune system is targeting. The adjuvant component of a cancer vaccine is really what stimulates immunity to your antigen target. The vector is the delivery system. It's how we deliver the information to the body to target that antigen. And the mode of administration is really exactly how that antigen target is delivered into the body. So it could be injection, gene gun, infusion, or a nasal spray. Now, what I'd argue and talk about today is the types of cancer antigen targets. There are tumor-associated antigens, and these are a class of genes that are really overexpressed and represent on tumor cells. So you're really telling your body to go after this thing that's highly expressed on a cancer cell. It may or may not be important. Tumor neoepitopes are a newer class of antigen target. Those are mutated pieces of proteins that are different in the tumor in comparison to the rest of the body. So it's a little bit easier to get immunity against those, but again, they may or may not be important. Another class I'm going to talk about is really the tumor driver and resistance genes. Now, these are genes that play a key role in tumor growth, metastasis, or resistance to therapy. And these can overlap with tumor-associated antigens or tumor neoepitopes. And we think this is really the key for cancer vaccines. And really, to kind of start a lot of our work, we've been focused on HER2-positive breast cancer. Now, I don't think I need to review it with this audience, but HER2 is really a driver and a resistance target for breast cancer, and it's overrepresented about 10 to 100-fold in this type of cancer as comparison to normal breast tissue. So it's sort of a tumor-associated antigen, and it's also a driver resistance gene. So in initial studies that we wanted to conduct, we used an engineered mouse, and this mouse develops HER2-positive breast cancer, but it also expresses another gene, green fluorescent protein. So it's a tumor-associated antigen, the green fluorescent protein, and also the HER2-positive gene. And we can immunize mice against HER2 or GFP and then determine which vaccine works better. We were able to break tolerance in these mice, and this is just using viral vectors. And this just illustrates we can elicit both T-cell responses and B-cell responses. So this is really the left and the right arm of immunity. So we can use these viral vectors to stimulate these responses very well in mice. And we've tried a lot of other methods, and we found viral vectors to be probably the most potent. So when we vaccinated mice at an early time point with our HER2 vaccine, we conferred significant anti-tumor immunity. And these mice, really, a lot of them were tumor-free. The tumors completely regressed. When we used a control or we didn't treat the mice, they had mostly developed tumors and the tumors progressed. But what was interesting to us is when we vaccinate against GFP, and GFP is really expressed only in the tumor cells, it didn't really have much benefit. So I think the key message here is that the type of antigen is really important, and going after something that's important for the cancer is probably our best bet. We've actually taken this viral HER2 vector into clinical trials, and this is just a phase 1 study. And what we found was that in patients with metastatic HER2-positive breast cancer, we could elicit T-cell responses in some patients and also B-cell responses in some patients. And what we found was that those patients where we could get a very good T-cell response, a memory T-cell response against HER2 specifically, those patients had a better progression-free survival, demonstrating some clinical activity of this vaccine, even in advanced patients. But a lot of these patients didn't really respond very well. And part of the reason may be that their tumors are highly immunosuppressed. So another tactic that we have with cancer vaccines is to employ immune checkpoint blockade. So this is just data showing in our animal models that they have high levels of PD-L1 expression in tumor cells and myeloid cells of advanced HER2-positive breast cancers. And so what we did then was we combined our HER2 vaccine with a PD-1 blocking antibody. And at the bottom, you can see that when we didn't treat mice or we treated them with a GFP virus, again, with these advanced cancers, there wasn't much benefit. PD-1 antibodies alone or the HER2 vaccine alone conferred some benefit, but in these advanced cancers, it wasn't a very significant benefit. However, when we combine these things, we got our maximal benefit. In a lot of mice, we had complete tumor regression and long-term survival. And so we're doing now a phase 2 trial at Duke that we're currently enrolling to test this approach. So in a quick summary, I just want to say that cancer vaccines, we think, are a promising area of development in the fight against breast cancer. There are lots of different options for cancer vaccines. I would argue that antigen choice is really important in this strategy, and that immunosuppression may require in advanced cancers that vaccines be combined with other types of therapy. And so I'd like to thank you for your time and attention, and I will hopefully talk to you and take questions after the break.
Hello, thank you all for joining us today. I'm Susanna Garcia, a postdoctoral research in Peru lab at UNC Chapel Hill. First, I would like to thank you, Susan G. Komen, for giving me the opportunity to share some of our science to this audience today. Our project related to metastases is entitled FG44 Regulates Tumor Subtype Differentiation in Luminal Breast Cancer and Metastatic Disease, and I'm going to tell you a bit more about it in the next presentation. Let me give you a bit introduction about breast cancer. I'm sure that you already know that breast cancer can be divided into three main clinical groups called hormonal receptor positive, HER2 positive, and triple negative patients. And these groups can also be classified based on the molecular subtype, being luminal A and HER2 enriched, the most common subtypes in hormone receptor positive, HER2 enriched and luminal AB also mainly present on HER2 positive patients, and basal like 80% of the time in triple negative patients. Based on this and other characteristics, the physicians choose the most appropriate treatment strategy. Despite progress toward early diagnosis and advances in treatment, patients with breast cancer experience relapse that includes distant metastases. Therefore, it is necessary to know which molecular alterations are involved in the metastatic progression and which of them could be modified by pharmacological drugs. So our research in the lab is focused on finding new target or specific therapies, not only on the primary tumor, but also in those tumors that may have spread to other parts of the body. These metastatic tumors could respond differently to the treatments than the primary tumor, because during this spreading process from the breast to other organs like lung, liver, or bone, the tumor can change its behavior. These metastatic tumors need to get adapted to the next environment that is a different organ from the breast tissue. Therefore, one of our goals is to compare what are the main differences between these primary and metastatic tumors, looking at the gene level and how these tumors change across time, or why these changes make them resistant to the standard treatments. In particular, we have studied 85 pairs of primary metastatic patients, where we have included all these different types of metastases and compared them to the primary tumor. After doing some analysis, we have found a potential target, a gene called FG44. This gene could be involved in the tumor growth and maybe in its ability to migrate through the body. This gene is present in some primary tumors, as you can see here in purple, and in different percentages in these three clinical groups. But most importantly, based on our findings, the gene level increase substantially when these tumors have colonized the distant organs. So in the metastatic tumors, FG44, represented here in purple, is more expressed, is more present than in the primary tumors. If we compare the subtype change rate between primary and metastatic tumors, we show that the luminal A had highest conversion rates, and offer some luminal A tumors here in blue change to luminal B that increased, and HER2-enriched tumors that increased compared to the primary tumors. Meaning that some primary tumors that were luminal A in the primary loses their luminal A characteristics in the metastatic setting and behave more like luminal B or HER2-enriched tumor, being more proliferative and having more aggressive phenotypes. The basal-like group, on the contrary, it's very stable and it doesn't change the phenotype between primary and metastases. These findings are important. Why? Because the treatment decisions are mostly based on the primary tumor characteristics, and in the area of precision medicine, treatments based on the metastatic biopsy could better optimize current treatment decisions. This is an example, and besides finding that FG44 levels are higher in metastasis tumors compared to the primary tumors, we also found other gene levels difference when we compare the immune cell content between primary and metastatic tumors. If we compare the gene level, for example, from an immune cell type, Bcells, between primary and metastasis in the luminal group here on the left, we can see that the immune cell content remains similar between primary and met. The average remains similar in luminal A, luminal B, and HER2-enriched tumors, but decreases in basal metastases compared to the primary tumor. And we have seen that this happened in the vast majority of immune cell content. So there is a difference in immune cell content between luminal or basal metastases. Why this is important? This could help optimizing immune therapy strategies in the metastatic setting. On the other hand, we also found that tumor proliferation or cell division rate increases in the metastases compared to the primary in the luminal set, in luminal A, B, and HER2 enriched, but it doesn't change when we compare primary metastases in the basal group. For now, we are using different approaches, such as mouse models, to test if some drugs responsible of blocking FGFR4 gene could decrease the tumor growth or change behavior in the primary or metastatic site. This is also an example of the tumor model that we have used. This tumor comes from a patient, a breast cancer patient, and it's been introduced in a mouse, and we call this a patient-derived xenograft tumor. This tumor is called WIM11 and is clinically ER-positive, HER2-negative, and presents high levels of FGFR4. It's also important to explain that this tumor was isolated from a patient with a fulminal clinical course and little evidence of sensitivity to endocrine therapy. So this WIM11 tumor is clinically ER-positive, but also reflects those cases of human breast cancers where FGFR4 may be critical. To determine if FGFR4 was a driver in these tumors, we use two possible druggable targets. We use FGFR4 blocking drug for blocking FGFR4, but we also use dual reversible blocking agent called lapatinib that it blocks HER2 and EGFR, noting the efficacy of this drug in the group of patients that are HER2 negative disease. That is also been demonstrated before. So these experiments using a mouse model also will help us to understand better if the gene can be targeted with a drug. How the tumor responds. You can see here in this map that the drug stops the tumor response. This here in light purple is the tumor treated with the drug over time, and it's much lower compared to the control, to the untreated tumor. Also looking at the gene level, we can study which are the future biological implications of blocking the gene, creating a group of genes that better represents the activity of FGFR4 gene, and we call this FGFR4 induced signature. We also can use a recently developed technique called single-cell sequencing using the lab. This is a powerful new tool for investigating tumor evolution and diversity in cancer cells, but very important in a single-cell level. As you can see as an example, there are three different situations using single-cell method. When the tumor isn't treated, when we treat the tumor with a FGFR4 blocking drug, or when we stop the treatment and let the tumor grow. Each dot represents a single cell coming from the tumor, and each color is a different population with different characteristics forming this tumor. So we can see here how these populations decrease. For example, all these populations here decrease when we treat the tumor, and other populations are selected. These techniques will allow us to understand the main difference between primary and metastases, the heterogeneity between these tumors, comparing separately the tumor cells, individual cells from the tumor, the individual cells from the immune cells, and the individual cells from the cells that supports the tumor. Nowadays, there are no breast cancer clinical trials designed with a selective FGFR4 or pan-FGFR4 inhibitor. So it's our hope to fully or partially describe the role of this gene in the preclinical setting, and in the best case scenario, set the stage for the next generation of clinical trials. We will keep working and hard to get the best results on this. Finally, obviously, I want to thank my mentor, Chuck Peru. He's the PI of this lab, and all the people implicated in our lab to help to this process. Of course, I want to thank Lisa Carey and Patty Spears for all the resources and guidance provided through this project, and of course, Susan Komen that has supported this study, and all the patients that contributed in this research. Thank you.
Wow, that is so inspiring, thinking about how immunotherapy could make a really big difference. I would like to thank our sponsors one more time, our local partners, Blue Cross Blue Shield of North Carolina and UNC Health, and our national partners, Seattle Genetics, Pfizer, Lilly Oncology, and Walgreens. Thank you for your commitment to finding the cures. Thank you again to Dr. Crosby, Alina Hamilton, Zachary Hartman, and Susanna Garcia for presenting today, and of course to my good friend Katrina Cook for moderating the Q&A. Thanks also to Patty Spears, a Komen advocate in science, who has been transcribing the answers to the question and answer and will post them all later today. Again, thank you for attending, and this concludes this part of the presentation.